So now we're going to switch gear to learn more about the virus from Dr. Erin DiCaprio.
Can you see my presentation?
Yes, I can see your presentation.
Wonderful. And obviously you can hear me. So welcome everyone. I'm Erin DiCaprio. I'm a virologist and food safety specialist in the Department of Food Science and Technology at UC Davis. Since my specific expertise is on viruses, I thought we'd spend a bit of time today comparing foodborne viruses to SARS-CoV-2, the virus that causes COVID-19. In my role as a food safety specialist, I also provide a lot of training and support for food processors in California. So if you have any questions at the end related to some of those regulations that Abby was talking about, I'm happy to address those as well. There we go. Okay, so I start most of my talks about viruses with this question, what is a virus? Because it's really relevant to the impact they have on food safety. Viruses aren't considered to be living organisms. They're obligate intracellular parasites, meaning that viruses require a very specific type of host cell in a specific organism to be able to multiply or increase in number. This is a really good thing, actually, when it comes to food safety, because viruses cannot increase in number in foods. Viruses also don't have any type of energy generating mechanism on their own. And this is another thing that differentiates them from living cells. Viruses are essentially just rationally designed combinations of nucleic acids, proteins, and sometimes lipids that have the ability to modulate a host cell's own machinery in a way to make additional viral copies. There are two basic structures of viruses, non-enveloped and enveloped viruses. I bring this up because these viral structures are very important in understanding how the different types of viruses persist in the environment and in foods. So non-enveloped viruses are the most simplistic of the two types of viral structures. Non-enveloped viruses are comprised simply of a viral nucleic acid, which is coated or encapsulated in a viral protein. The viral protein that encases the nucleic acid is called the viral capsid, and the viral capsid is formed from a protein that's encoded in the virus's own genome, and the capsid can be comprised of one or several different viral proteins depending on the virus. The genetic material of viruses can be either DNA or RNA depending on the viral type. So that's another unique characteristic of viruses compared to living cells. Sometimes these viruses don't have to have DNA at all in order to infect cells. Envelope viruses are more complex in structure compared to non-envelope viruses. On the exterior of an envelope virus is a lipid bilayer that's derived from the host cell's own membranes. And within that lipid envelope is viral nucleic acid, and that nucleic acid is coated in additional viral proteins. Depending on the type of virus we're talking about, there can be many other viral proteins associated with that nucleic acid or in the space between the nucleic acid and that lipid envelope. Another very important structure that envelope viruses have are viral proteins that are associated with or embedded in that viral envelope. These are very important for allowing the virus to attach to host cells, to be able to fuse and enter those host cells. They're also very important for maintaining the integrity of that viral envelope, and at the very end of the viral infection cycle, they're important for getting new viral particles out of a host cell. So I introduced that because it's really important in understanding which type of virus is more stable. So the structure of viruses is very critical to the resistance to different treatments, including heat, UV sanitizers, but also their general stability in the environment or in foods. So to render a non-enveloped virus non-infectious, you can damage the capsid or the nucleic acid. An envelope virus can be inactivated by damaging envelope proteins, by damaging the envelope itself, through damage to other viral proteins, or damage to the viral nucleic acid. Generally, envelope viruses are much easier to inactivate compared to non-envelope viruses because that viral envelope, that lipid bilayer that covers the exterior of the virus, can be destabilized by many sanitizers, by surfactants, through mild heating, and also by drying out. These envelope viruses also have multiple targets that can render them non-infectious, including external proteins on the surface of the virus that can easily be damaged by different chemical agents or UV. The capsid of non-envelope viruses is generally more resistant to heat than the lipid bilayer of envelope viruses. Also, non-envelope viruses are much more stable under dry conditions compared to envelope viruses, and generally very harsh sanitizers must be used to inactivate non-envelope viruses. So there are many different types of microorganisms that are foodborne pathogens. This slide lists them from most resistant to least stable. This means resistance to heat, sanitizers, and desiccation. So prions are really our most resistant type of foodborne pathogen. Prions are pathogenic agents that are transmissible and induce abnormal folding of specific normal cellular proteins called prion proteins that are found primarily in our brain. The prion disease that everyone's probably heard of that's related to food is bovine spongiform encephalitis, or mad cow's disease. To inactivate prions, we have to apply very, very high heat or harsh chemicals to get rid of these prions. Mycobacterium, you see listed on this slide, this is a genus of bacteria that cause severe disease in some cases in humans. You've probably heard of Mycobacterium tuberculosis. These particular types of bacteria have a very unique structure, and this structure imparts a very strong resistance to sanitizers and heat. There are types of foodborne bacterial pathogens that have the ability to form bacterial spores that we know are very resistant in the environment and to sanitizers and heat treatment. However, normal growing bacterial cells, i.e. vegetative bacteria, are not very resistant to disinfectants or heat, although we know many of them can survive in the environment or in foods for long periods of time if conditions are ideal. So kind of correlating this back to viruses, non-envelope viruses, which include norovirus, hepatitis A virus, poliovirus, adenovirus, and many others, are more similar to bacterial spores and mycobacterium in their resistance to heat and sanitizer inactivation. And due to that fact, they remain infectious in the environment and in foods for long periods of time. Envelope viruses, in contrast, that include influenza viruses, coronaviruses, measles virus, and many others, generally do not survive for long periods of time in the environment, and they can also be easily inactivated by heat and chemical treatments. Listed here are the major food and waterborne viruses grouped by the type of disease they cause in humans. So all of the viruses listed here are non-enveloped and are transmitted by the fecal oral route. This means you have to ingest these viruses to get sick, and the primary site of replication for all of these viruses is the digestive tract. So they're actually actively growing in your digestive tract. Since all of these viruses are non-enveloped, they can remain infectious on surfaces, in food, and in water for weeks to months. So there's currently no evidence of transmission of SARS-CoV-2 via food or food packaging. So SARS-CoV-2 is the virus that causes the disease COVID-19. So this virus, SARS-CoV-2, is an envelope virus, and current data suggests it only remains infectious for at most a few days outside of the body. Also, the transmission mode for this virus is via respiratory droplet, meaning the virus must contact your mucous membranes either via aerosolized droplets created by someone with a COVID-19 infection, coughing, sneezing, talking, eating, etc., or by you touching a surface where these droplets have settled and then touching your own mucous membranes. So these characteristics make it unlikely that food would be a vehicle for a transmission of this particular virus. So there, when we talk about food safety, nothing is ever zero risk, but really at this point in time, the risk of food or food packaging being a vehicle for COVID-19 transmission is very, very low. So what can you do in your processing facility to help mitigate, the low risk that there might be of SARS-CoV-2 ending up in your food? The big one is to follow those existing GMPs in your food safety plans. Um, Make sure good personal hygiene practices are being followed. So washing your hands, excluding anyone that has symptoms of COVID-19 or suspects they may have been exposed to COVID-19, from working on the line or handling food. Um, at this point in time, I think really the risk is to our workers and not to the food supply. So it's really important that you take steps to try and protect your workforce. So implement social distancing as much as possible in your facility. I know that can be a major challenge with the way that, a lot of these processing lines are set up. The CDC actually just came out with a very nice guidance document for how to implement social distancing in a processing facility. It's targeted more towards meat processors, but I think a lot of the information can be broadly applied to food processors in general. So I make sure that I have a link to that put in the chat. Additionally, some processors are, employing face masks to help minimize unintentional virus shedding from someone that might be asymptomatic in order to, help protect other workers in that space. A lot of processors are also implementing disinfection of high touch surfaces in the facility, such as, door handles, handles in restrooms, break room tables. So this can help you reduce any indirect transmission of the virus in your facility. So really, kind of as was reiterated in the previous two presentations, really rethink, what you're doing and think about, where in your facility would your workers be put at the highest risk of being in close proximity to others. Think about those surfaces in the facility that are going to be contacted by multiple people throughout the shift, and take the steps necessary to help protect your own workforce during these really challenging times. So next week we'll talk a little bit more in depth about, cleaners and sanitizers and disinfectants if you're going to use them in light of what's going on with the COVID-19 pandemic. We've seen some uptick in misuse of chemicals by consumers, and so we just want to make sure that everybody is on the same page with, what they should be doing in the processing environment to help mitigate any risks that might be there. So I didn't share here, but I co-host a website called UC Food Safety. If you plug that into your browser, it should come right up. And we have been kind of developing a repository of all kinds of COVID-19 food safety related guidance documents and other tools that have been developed by, people within our university, but then, at universities across the country, also trade organizations and the regulatory bodies. So I encourage you to check out that website and see if there's anything um, that helps answer any questions for you. So I'm happy to address, any COVID-19 questions during the Q&A session, and then also help with any California specific processing questions that you might have.